Alrighty. Well, hello everyone. How are you on this wonderful evening right now where I am, which is the true time? Today, I've got on the wonderful biologist, Matthew Herron. So,
Nice to be
hi.
here.
So, uh, I have some questions from him, and would you be interested in any questions from the chat, or you want to just proceed with the questions?
Sure, yeah, that sounds like fun.
Okay.
So
Uh,
you're live streaming this then, I take it. I just
okay.
lost audio.
Okay. Uh,
I
so,
lost audio there for a second, so if you
Oops.
ask me
Sorry about
something
that.
before I
That's
lose
my
your
fault. That was my fault.
okay.
Audio is kind of weird on my end sometimes. I live in Shreveport, Louisiana. It's not the best, especially where I am. So, okay. So, please, for anyone who doesn't know, describe your background, research, and all that sort of stuff.
I started in biology a little bit later in life. I'd been out of, can you hear me? Okay, I see you're muting your mic.
Oh yes, yeah, I can hear you. You
Okay.
sound just fine.
All right, I just saw the mute thing and yeah, so yeah, I came to biology a little bit late. I did my undergrad was actually in political science, and then it took me a few years out of school before I decided to do a master's degree in biology. And I did that at the University of Central Florida, studying mostly squirrel phylogenetics. And then I went on to a PhD at the University of Arizona with Rick Michaud, who was studying Volvox. And that's where I really started getting interested in problems related to the evolution of multicellularity and really got enamored of the Volvocene algae as a model organism for that problem. I actually left that for a while for my first postdoc at the University of British Columbia, where I was working with a mathematical modeling guy named Michael Dobley. And that, well, I won't give you the whole story, but long story short, I met my second postdoc advisor at a conference in England, and he convinced me to apply for a NASA fellowship and basically what I wrote that fellowship proposal to do was the experimental evolution of multicellularity in the unicellular green alga Chlamydomonas, like you were referring to earlier. And pretty much I've been working on that ever since. I still do some comparative stuff as well, which is really what I did my PhD on and never really got away from. And some theoretical stuff as well. But most of it is related to the evolution of multicellularity and or the major transitions more broadly.
Well, that's pretty cool. As it happens, I was looking up some of your publications earlier. I typed in your name, and I did see the Sayerd paper, and I totally forgot to jot that down in my notes. But yeah, I thought that was kind of funny.
I was just at a talk today and I got all excited because they had a picture of Xeris, which is that's basically what I did my master's degree on, this African ground squirrel called Xeris.
Oh yeah,
Yeah.
that's pretty cool. Uh, we just have a bunch of gray squirrels where I am. I was down at uh, LSU Baton Rouge, and I got to work in the or intern in the Department of Mammalogy, and
Oh,
so
cool.
I got to handle a lot of squirrel skins. That was interesting.
Yeah.
But yeah, squirrels are neat. I've uh, you ever hear of like Dr. Esselstyn?
It doesn't ring a bell.
Uh, so, so yeah, you do a lot of work in the origin of multicellularity. So does that? And you said that primarily revolves around Chlamydomonas and Vulvacenes and guys like that. Do you ever do work on like the origin of like metazoan
No, not really.
multicellularity? Okay.
No. I think the most exciting work that's being done on that front is, you've probably heard of this, is Nicole King's work with choanoflagellates. Nicole King
Mhm.
and basically people who have come out of her lab. Um, choanoflagellates are the closest known unicellular relative to animals, and so they're a great model system for those kinds of questions. And they found some really cool stuff in that system. but that's not what I work on.
Yeah, I think it's the evolution of Volvocines that was happening like what 200 million years ago, that kind of stuff.
We think, yeah. I mean, those estimates are, there's pretty big error bars on those estimates.
So for anyone who doesn't know, that's like a late Triassic area. Uh, but yeah, I mean I think that's really cool because you've got, I mean, there's this whole range. You've got on the one end like the little two-celled gonium, who are going around. You've got like is it Tetrabana, which is a
Yeah,
four-cell.
Tetrabena is usually 4 cells, you know.
So so do you think that there's like a particular reason that they associate? Like, do you think it's primarily predation or what?
That's really the question. I mean, there's been a lot of proposed advantages to multicellularity itself, to basically having more than one cell, and size is definitely one of those proposed advantages. Just really, there's a lot of proposed advantages that are centered around size, and predation is one of those. Just the idea that if you get too big for a lot of the little filter feeders to eat you, then you've got a big viability advantage. There's other ideas that are related to size as well. Things like homeostasis may be easier in a larger group, and resistance to environmental insults like sudden changes in temperature or toxins or things like that. When you actually have an inside instead of just basically your whole cell being exposed to the medium or to the environment, then you can imagine that in the case of some major environmental insult, you're buffered against that if you're on the inside of some kind of multicellular structure. So that may be an advantage too. There certainly are disadvantages. We know that because multicellular things tend to reproduce more slowly than unicellular things. And there's a pretty well-supported inverse relationship between size and, or I guess it's a positive relationship between size and generation time. So there's that built-in disadvantage. So, we think there has to be some substantial advantage to outweigh that, or else it probably wouldn't happen as many times as it has. But yeah, certainly predation is one of those ideas, and that's one that we're looking at as a possibility.
Yeah. Would you like to tell them about your experiments with the paramecium and chlamydomonas?
Yeah, sure. Basically, we set out to test this idea that predation would drive the evolution of multicellularity. And so what we did is we just co-cultured this unicellular green alga, chlamydomonas, which is a relative of multicellular algae like Volvox. We co-cultured them with this filter-feeding predator called paramecium. Paramecium is pretty good at eating single-celled chlamydomonas, but they can't eat efficiently things much bigger than that. So we hoped that, if some mutation came along that caused them to form a multicellular structure, then that mutation would likely go to fixation. It would be selected for because they would have this viability advantage. And I guess I can talk about this because we have a preprint out about this. It's not yet peer reviewed. We found in 2 of the 5 experimental populations that multicellular structures did evolve, and we didn't see that in any of the control populations. So what we think this shows is not that predation was the driving force behind any particular origin of multicellularity, but just that it is a plausible selective pressure that can drive the evolution of multicellularity. To know that it had happened that way in, say, Volvox, we would have to have a lot more information. We'd basically have to have a time machine or some kind of evidence I haven't thought of yet to test that idea.
Right. I think similar experiments have been performed with Ochromonus.
Yep, and
The
with chlorella, as a matter of fact.
chlorella, yeah, that's
Yeah.
what it was. Yeah, I think that's all
I
like
think I
really.
think I think you're right. I think Ochromonus is the predator, and chlorella is the algae. Yeah.
Yeah, it's just such fascinating uh, experiments. Uh,
Thank you.
Also, oh, and again, for anyone who just arrived, if you have questions, you can put them in the chat, and so we'll try to get to them. Uh, so as far as the phylogeny of volvocines and their relationship to other uh, organisms, can you speak on that a little bit for us, please?
You want to know about the
Yeah,
phylogeny within that group, or how they relate to other
yeah,
things?
within that group, and also how they relate to others. If that's all right.
Okay. Yeah. Well, their phylogeny is actually really interesting because they uh, they seem to have a lot of convergent evolution. There seems to be several lineages that have basically the same body plan. So, for example, the I'll call it the nominal genus Volvox because we usually don't think that polyphyletic assemblages are good taxa. But the group of things that are called Volvox actually probably have at least three separate origins from smaller algae. So, they were originally described as the same genus because they were described using, their morphology, using what they look like. And they all do look pretty similar. They have some important developmental differences. And actually, there were, some indications even before molecular techniques came along to work out these relationships that these things that were included under this umbrella of the genus Volvox might not all be that closely related to each other. And when the molecular phylogeny started coming out in the 80s and 90s, it was shown pretty convincingly that, in fact, these things were not, at least some of them were not closely related to each other. In fact, some of the divergences between members of the nominal genus Volvox go back almost to the base of the Volvocine tree, which, as you alluded to earlier, is something on the order of a couple 100 million years ago. So it's been a pretty amazing example of convergence in that group. As far as what they're related to, um, they're they're nested within a big clade of unicellular green algae that mostly look pretty much like Chlamydomonas. Chlamydomonas is another, at least paraphyletic assemblage. Uh, there's hundreds of species that are called Chlamydomonas, and I'm hedging by saying things like they're called Chlamydomonas because in reality they need to be taxonomically rearranged into probably several different genera. And some people have actually tried to do that and have set up, I think they've actually set up new genera, but this has all been really recent and it's not yet clear whether people are going to follow the new taxonomy. Anybody can propose a new taxonomy. It's really just a matter of whether people believe it or agree with it, that'll determine whether they follow it or not, and whether people start calling these things by these new genus names. And then that group is, these are all members of the Coryphaceae, which is one of the two major groups of green algae, the other one being the Ulvophyceae, the ulvophyte green algae, which includes things like sea lettuce. And then that group is closely related to land plants. The green algae, specifically the charophyte green algae, are the closest living relatives of land plants. And then the sister to all of that, to the green algae and land plants, is the red algae. And those things are all in the eukaryotic supergroup that descends from the original acquisition of the chloroplast. They call them the archaeplastida. Basically means ancient chloroplast or ancient plastid. Because everything that's not in that group that is photosynthetic, I'm talking about eukaryotes here, became photosynthetic by consuming a eukaryotic alga, a green or red alga, or so that's considered a secondary endosymbiotic event. And then there are actually tertiary and quaternary endosymbiotic events where these things get eaten by other other organisms, and they steal them and become photosynthetic. So things like brown algae, for example, don't descend directly from this original acquisition of the chloroplast. I forget if they're a secondary or tertiary endosymbiosis.
Yeah, I remember reading about was it like the chlorarachnophytes, whatever, a group that have like you have like the red algae who have their plastids, and then the chlorarachnophytes take in the red algae, and so it's like the
Yeah, exactly.
successive tiers of eating each other.
Yeah, it's like and I'm not the first to describe it this way. It's like a Russian doll configuration in some of these, where the original cyanobacterial cell, let's call it, but is now a plastid, is like 4 layers deep within a eukaryotic cell.
I think John Archibald has done some research on like the nucleomorphs, aren't they? Like the vestigial remnants of a nucleus that are still within like some of these organisms that have done like tertiary and quaternary
exactly
events.
Yeah.
Which is crazy to think about, because like here it is.
Yeah, that may be, we talk about multicellularity as being probably the most common major transition, or transition in individuality, but that's probably another really common one that seems to have happened lots and lots of times across the tree of life, these endosymbiotic origins of chloroplasts.
Yeah, it seems like because you look at work by like was it Thomas is it Cavalier Smith?
Cavalier Smith,
yeah, because they're like always doing different trees of eukaryotes, because they're like, oh hey, it turns out this is actually convergence, or this is a HGT or something like that, and so this is rapid evolution right here, and so it's always like changing a lot, because it's hard to figure out is this natural to this clade, or did they steal it from this other clade? How did that over there? And there's just it hurts my head thinking about how much literature there is on that.
Yeah, it's immense.
Just looking at things like what was back in the 70s or whatever, the Archaeozoa, the group was like but they were like the Archaeozoa, like they had like highly modified mitochondria, like they modified them into mitosomes and things like that, and so they were thought to be like pre-mitochondriate eukaryotes. But it's like no, they're not.
They're secondarily lost the mitochondria or reduced them.
Yeah.
You talk about this huge literature, we talk about it, we just say, there's been tons of these secondary and tertiary and quaternary endosymbiotic events, but each one of those was an immense amount of work by some research team to piece together that story and figure out, oh, this thing used to be a red alga or whatever.
Yeah, absolutely. Yeah, I agree. There's a whole lot of work that goes into that. Right. And so when some of us over here on Twitter and YouTube and whatnot try explaining some of this stuff to people who are somewhat resistant to the ideas, it's a little bit difficult, because they don't realize how much research has actually gone into these fields.
Yeah.
And it instantly becomes an arduous task trying to even like briefly explain this field of study. It's like, just go to Google Scholar and type in this term and look at all the results.
16,000 hits.
Yeah, exactly. Yeah, suddenly it becomes a little bit difficult. But I do have one question from the chat. Have you heard of Arn Ra's phylogeny project?
No, I don't think so. Not by that name anyway.
Okay, I'll send you some information on it afterwards. So, yeah. So there's that. Speaking of people resistant to the ideas, I mentioned before we got on you have bumped into some ID proponents. Is it just ID proponents, or have you managed to meet creationists too, or just them not at all?
I haven't met any of them. I've only, interacted with them online a little bit. I don't really think I've messed with the young Earth creationists too much, or if I have, it's been below their radar. My web presence is, I think, too small for a lot of these groups to notice. But yeah, I've annoyed members of the Discovery Institute a couple times.
Like Ann Gager, for one.
I don't know if she ever noticed what I wrote, but yeah, she. It was actually right when I had first. It was really fortuitous timing for me because I had just set up my blog, and she published a thing that was, just right in my wheelhouse. She was talking about the evolution of multicellularity in Volvox. And it's like, well, I can't let this go by. I gotta, and of course it was just completely full of, misconceptions and, the frequent thing that you see with a lot of ID creationists is they have a certain conception in their head of what evolution is, and it's mostly wrong. And then they look at real scientific studies and they see that they don't match up with the idea of evolution that they've got in their head, and then they say, well, so evolution must be wrong. And so, you know, she was making that mistake a lot, and also just getting the facts wrong quite often. I think one of her arguments was that there must have been an immense number of new genes to get from a unicellular alga like Chlamydomonas to, a complex, differentiated multicellular organism like Volvox. But we know empirically that's not true, because both of those genomes have been sequenced and they have practically identical gene content. So there's been very few new genes between the common ancestor of Chlamydomonas and Volvox and the modern species. So yeah, that was fun. And then there's been a few other times where it's such a fire hose of misinformation that I really can only pick at it every now and then. And usually when it's something that's either just verifiably false in a really simple way, something you can just look and say, no, the data show that that's actually not true. Or when they've contradicted themselves, which does sometimes happen, or when it's just directly in my wheelhouse, because they really do pretty much comment anytime there's a new finding about the evolution of multicellularity. And of course, that's just, like I said, right in my wheelhouse. So I usually have to hit back when they say stuff about that.
Oh yeah, definitely. Yeah.
But like I said, my blog is pretty small potatoes, and so I don't think they usually even notice when I do that, or if they do, they ignore me.
Oh, they notice quite a bit. I mean, when I was working for Jake Esselstyn down at LSU, I actually found out that they had written an article on one of the species of shrew rats he had found. And they quoted him they wrote his name was actually in the article, and they were like, it's just the same kind of rat, it doesn't matter. And I showed it to him, and he was like, basically said, I don't care.
Yeah, rats are rats, right?
Exactly.
I did manage to get myself banned from one of their blogs, the one that does allow comments, but that was really it really didn't have much to do with intelligent design. It was because I reminded one of the authors that he had previously called me forget what the exact wording was, but it's basically a fascist sympathizer because I pointed out that he had lied about something.
Good Lord, a fascist sympathizer. Goodness gracious.
Well, that wasn't the exact wording. I can't remember exactly what it was.
I mean, that's pretty crazy. Well, I mean, if you're an evolutionist, then you're a Marxist, communist, whatever.
Right.
I mean, you look at was it Jonathan Wells is like, evolutionists believe everything. You can be, from just evolution to like social Darwinism, eugenics, Nazism. It's like everything is just thrown under the label of Darwinism or evolutionism, whatever, because it's so much easier to fight, a monster when you put everything you hate in it. So
Sure. Yeah. I mean, straw manning is one of their favorite tactics.
yeah. Oh yeah, absolutely. Yeah. I 100% agree with you, on these things that are just easy to look up factual errors, but as RJ Downard and I know, and I'm sure lots of other people who interact with them on Twitter. Also, fun fact, Beach Price says you're now in her hero book. So
for getting banned. Well, it's not that hard.
I've been banned by Ken Ham now, so unfortunate. I can't see his posts anymore on my regular account.
On Twitter.
Yeah, on Twitter. Yeah. I went back one day and I was just like, "Oh no, I've been banned." I poked him a lot. So
I do want to say, unlike Evolution News and Views, Uncommon Descent allows comments, and I have to say I respect that because they do get a lot of pushback. Pretty much, I think pretty much every article they post gets some pushback in the comments, and I respect that they allow that. They don't block everybody who disagrees with them, but
yeah,
I don't think it's that difficult to get banned.
Yeah, I agree, and a whole lot of what they post is, as you said, easily correctable. Is easily able. I mean, you don't even have to go to Google Scholar. Just type in like Wikipedia. Just go to Wikipedia and say, "Hey, what's this?" and you'll get a list of results. And then, of course, they'll link you back to the technical work where you can go check that for yourself, their methods and materials and all that. But they just flat don't do that. And sometimes you have to wonder, is it like, what's going on in their brain that is preventing them from doing 10 seconds of Google searching? What is it?
Yeah. I mean, I don't think that's true of all of them.
I agree. Yeah.
Evolution News and Views tends to be a lot better argued than Uncommon Descent. Uncommon Descent often fails to even make a coherent argument, and I don't mean like a logically sound argument. I mean just an argument you can even follow. It's just oh, here's this new thing, so obviously it must be intelligently designed. But Uncommon Descent, or sorry, Evolution News and Views, most of those guys, they're familiar guys and women. It's not all guys there. They dig into the primary literature. They clearly follow. Even I've seen them blog about preprints on bioRxiv. So they're clearly following the primary literature and some of the secondary literature. They just have a certain filter that they read everything through.
Yeah, that's one of the I think the most interesting person as far as like anti-evolutionists go is Georgia Purdom.
I don't even know her.
She's a geneticist, like an actual geneticist, has done work in like a cancer research, I think, something like that. So has a real PhD and all that. She's a young Earth creationist, and she's on like the top tier of answers in Genesis. And it makes you wonder, because I've actually read her work with regards to mutations and genetics and things like that. And it's laughable. She cites like 2 or 3 technical papers and then misquotes those. She'll take a single sentence out of it and be like, aha, we got them. But then if you look at the whole next paragraph, it's like, here's why it doesn't say what she thinks it says or wants
Yeah.
reviewers to think. What do you think of that?
There's a certain line out of Darwin that gets used for that a lot, which is, and I'm not going to be able to quote it, but where he's talking about the eye, the like the vertebrate eye, and he basically says, it's really hard to imagine how an eye could evolve through stepwise, small changes, each one of which has to be adaptive in its own right. And then he goes on to explain how it could have happened in a stepwise fashion. And there's comparative, data that show that what he's saying is at least plausible. He goes into one or more paragraphs describing how this could have happened in a stepwise fashion, where every step was adaptive. And that's almost a test to whether you're dealing with somebody who is at least arguing in an intellectually honest way. If they just quote the first sentence where he says how hard it is to understand how this could happen, and don't go on to say that Darwin then gave a plausible explanation, then they're being intellectually dishonest, and you do see that now and then.
Oh
You
yeah,
you don't
absolutely.
see it much. I don't think I've ever seen anything that blatantly dishonest on like evolution news and views. But those are the guys that are really. Most of the people writing for that blog are some kind of fellow at the Discovery Institute.
Mm-hmm.
So they're people who I don't actually know. Like, are they doing this for a living? Do they draw a salary from the Discovery Institute? I don't know.
I don't know either. I mean, uh, I know some of them have published. A number of them have published actual research, like Wells and I think Gager and a few others.
Sure.
But I don't know if I honestly have no idea if they do that for a living. They might. I could believe they could, attain a salary putting out these papers to their subscribers, and get money from that. So I don't know. RJ, do you know? I know you're in the chat. Do you know if they get paid? So we'll see what he says on that. But that's a good question.
It's probably a not-for-profit organization. They probably have to file, tax returns that show where the money's going.
Yeah.
It's not, if they're getting paid for it, fine. I mean, there's nothing wrong
Oh
with
yeah,
that.
absolutely. He says most of the hangers on are not paid. There may be 20 operating fellows,
So that sounds like the fellows do draw a salary.
I guess. So yeah. Interesting. I do have another question to bring it back to multicellularity. I do have another question which I forgot to ask earlier, but I keeps getting my brain, then I lose it and it comes back. I want to ask you about how do or if we do know at this point, how do organisms go through from a colonial phase to the unicellular phase and ontogeny, where they develop into a multicellular organism? Has that been worked out?
Sorry, can you repeat the question? I'm not sure I understood it.
So like how multicellular organisms have to go through a unicellular phase in their ontogeny. How do
Sure.
we go from the colonial, where it's just these organisms associating, to where they get to that point where they go through that stage in their development? Has that been
Well,
worked out?
it certainly hasn't been worked out thoroughly, because
Okay.
I mean, the question you're asking, I would describe as the field of Evo Devo. So, but there is a lot we do know. One of the important things, and I think, you called it a colonial stage. To me, colonial basically means a multicellular structure that lacks cellular differentiation, that lacks coordination and communication among cells and things like that. I consider Volvox, for example, to be a multicellular organism where you might fairly describe something like Tetrabena that you brought up earlier, these four-celled relatives of Volvox. You might fairly describe them as colonial because there's not evidence that the cells really communicate. There's not any complicated developmental process. There's no evidence of cellular differentiation. It just looks like four basically identical cells that swim around together in a clump. So, how do you get from that to a differentiated multicellular organism with maybe a more complex developmental program, or ontogeny, as you called it? That's a tough problem. That really is the field of at least developmental biology. When you're doing it in a comparative sense, it's evo-devo. In the group I study, David Kirk, who actually just recently passed away, he was sort of the grandfather of Volvox research, and he was active until just a couple of years ago. He published a paper in 2005 that he called a 12-step program for evolving multicellularity and I think cellular differentiation. I may not be quoting the title exactly right, but essentially he did a really similar thing with the Volvocene algae to what Darwin and some later authors did with the vertebrate eye. He looked at extant species, of which there's a huge range of sizes and degrees of complexity, and he constructed sort of a plausible series of steps that could lead from a unicellular ancestor to a differentiated multicellular organism like Volvox. And so, some of the important steps are things like just staying together. That's, we think that's the first step. You've got to form a structure of more than one cell to be multicellular. Then, really importantly, the group that includes Volvox, let me turn off my ringer real quick here. The group that includes Volvox, that family, which is called the Volvocaceae, they all go through a process of inversion during development, where they start off being inside out as embryos with their flagella on the inside. Yeah, it's weird. And then they turn themselves completely inside out during development. And we actually know a lot about how that happens, about the genes involved and the sort of morphological processes that are involved. But, that seems to be a really important step. And, to me, that's a really sort of organism level process that suggests that things that do that are fairly considered multicellular organisms. There's some gray area here. We can argue about what's a colony and what's a multicellular organism, but and if you say, well, for me, it has to have cellular differentiation, I won't disagree with you. I don't feel strongly enough about it to argue about it. For me, I would consider something with inversion to be a multicellular organism. Another really important step seems to have been the expansion of the extracellular matrix. So when you look at a Volvox colony, it looks like a hollow sphere with all the cells I'm making hand motions as if you can see them. See now they're below the camera. All the cells are arranged on the periphery, on the surface, basically. The stuff that looks like it's empty is extracellular matrix, and it makes up most of the volume of these colonies. And then, of course, the really important thing is cellular differentiation, where Volvox has specialized reproductive cells and cells that are basically specialized for swimming. And again, we know a lot about the genetics underlying that distinction. It's basically a matter of differential gene expression. Because, like most multicellular organisms, all the cells in Volvox are genetically identical to each other, just like they are in our bodies, except for new mutations that happen during development. So, what distinguishes a somatic cell from a reproductive cell, or what distinguishes a liver cell from a muscle cell, for that matter, is mostly gene expression. They have differential patterns of gene expression. And that's actually been worked out pretty well. There's been a couple recent papers where people have um, done whole transcriptome sequencing of somatic cells and reproductive cells separately, where they actually separated two cell types, looked at gene expression in each one and compared, well, how are they different from each other? And they found that there's lots and lots of gene expression differences between those two different cell types, but this is one of the big messages of Evo Devo, is that even though there are thousands of gene expression differences between those two cell types, all those expression differences are caused by just a couple of genes, just a couple of transcription factors that control all that gene expression, directly or indirectly.
That's really neat. It's all very fascinating research.
On a more conceptual level, one of the things we think may be important in cellular differentiation is every organism on Earth has phenotypic plasticity. They respond to their environment in different ways. And that's true even of unicellular organisms. So that was probably true of the unicellular ancestor of Volvox. So even these single cells have the capacity to have, for example, different gene expression profiles, depending on what the environment is. And what we think is a really important factor is that those plastic responses get co-opted into cellular differentiation, at least the earliest forms of cellular differentiation.
Yeah. Have you ever looked at like the heterocysts in bacteria?
Like in cyanobacteria?
Yeah.
I haven't done that myself. I think it's a fascinating field of research.
Yeah, because they kind of have an almost like a sort of differentiation kind of going on there, you know?
Yeah, I would certainly consider it a form of cellular differentiation. And, I would consider, Anabina, for example, to be a multicellular organism.
Yeah. For anyone who doesn't know, basically this like cyanobacteria, they make these like structures where they have cells who are basically the ones making the materials and then like passing it on to the others, right? The metabolic materials that they need, they're kind of passing it to others. Would you say it's a good explanation of it?
Yeah, the way it was explained to me, I think, and it's been a while since I thought about this, is that nitrogen fixing, which is what the heterocysts do, is, at least in those bacteria, is incompatible with, I think it's oxygen. Of course, the photosynthesizing cells are producing lots of oxygen. So it makes sense to make a separate compartment where the nitrogen fixing can happen so that it's not poisoned by oxygen.
Right. Yeah, it's really cool to think about. Same with the also the work on multicellularity, or the weird little the dictyostelium, the little slime molds.
Cool.
That's also really weird to think about, because they aggregate when they form their little the sexual body, which is just bizarre.
Yeah, there's actually quite a lot of different things that do that form multicellular structures through aggregation, and they all seem to do basically the same thing. With clonal multicellularity, which is what we are and what plants are and things like that, most of the complex multicellular organisms that we encounter every day are clonally developing. But this aggregative development has evolved lots of times independently, and they all seem to do basically the same thing. They form a multicellular structure, then they form a fruiting body, and they use it to disperse their spores.
Yeah.
I've always thought it was kind of an odd, with clonal developers, we see a huge variety of things, of strategies, basically. Plants have a very different strategy from animals. That
Yeah.
happened to be strategy
Yeah.
from brown
A lot
algae and
less
stuff like
fun, I would imagine.
that. Yeah. But
But,
for some reason, all these aggregative developers all seem to do it for the same reason, or at least to do the same thing with it, with the multicellularity.
yeah. Yeah, it's pretty crazy. How many times would you say do you think has multicellularity independently evolved?
That is a great question. Uh, the number that gets thrown around the most is 25. I think that that is likely to be a big underestimate. And the reason I say that is that if you imagine what the earliest multicellular organisms in each lineage must have been, I think the two things that most of them probably had in common is that they were small and that they were soft bodied. And those are not traits that lend themselves to easy fossilization. So I think it's entirely possible that there are extinct multicellular lineages that have never shown up in the fossil record, or if they have, we didn't know what they were and we shoehorned them into some other group, or just called them small fauna or something like that, gave them some generic name.
Right. Yeah.
So yeah, I think 25 is a minimum. It could easily be many times that, I think.
Oh yeah. My hope is that things like Charnia and all and Cyclomedusa and the other really weird like Ediacaran fauna don't turn out to be like independent protists or something like that.
Well, I think that idea just got a big boost. That
Oh man.
It hasn't. It's a little bit outside of my expertise to evaluate whether what they're saying is true or not. But there was a recent paper that drew the conclusion that at least some of the Ediacaran fauna were animals.
Oh yeah. I mean, like Dickinsonia is an animal. It seems to be from its ontogeny, and it's like a bilaterian. There are a few other kind of sort of bilaterians, which they've grouped in this. Well, we don't really know. Some people call it like a proarticulata or something like that, because it's like we don't really know where they fit on the tree. They could be way up here or way down here, you know. And so, but yeah, I don't know. Some people are, like, oh, they're all animals, but other people are like, well, maybe a bunch of them aren't animals. Maybe a bunch of them are essentially like, these derived protists or whatever. But I kind of hope that they're not.
What we need is a time machine.
Exactly, we do. We need to go back and sequence some Ediacaran genomes.
Yeah, and some Precambrian and Cambrian genomes as well. We'd sort out a lot of the existing controversies pretty quickly, I think.
Yeah, that would be pretty nice. We'll get someone on that in the near future, I hope.
Please do.
So anyways, it's been almost an hour. So want to call it quits? Are there any other topics that you want to broach?
No, I really just came to see what you wanted to talk about.
Okay. And I don't really see any questions from the commenters really at the moment.
I do have a question. What's the poster there behind you?
That one?
Yeah. Poster or painting or whatever it is.
Yeah, that's Phylogeny of Life. I got that from Useful Charts. That's what it is. It's really nice. Yeah, Useful Charts. They do lots of them. They have one that's like the evolution of languages, the evolution of different religions. It's like this huge chart where they point to different ones. I got that one because, of course, I love phylogenetics, and so it's it's pretty good. Some of the polytomies kind of bug me. Like this one right here, it says life, then goes straight up and says eukaryote domain, then says plants, fungus, animals, all coming off the same little stem, and I went. But
maybe not the most up-to-date eukaryote phylogeny then.
Yeah, there are a few things I kind of applauded them on, like they have Atlantogenata way up there in the top where they put the elephants, and the other Afrothairs and the aardvarks and sloths and armadillos all. Not aardvarks, sorry, that's old. Anteaters and sloths and armadillos and elephants, they all come together and they're their own little branch. So I give them props for that one. But yeah, there's so much research on it's like phylozoans and holozoans and all those guys, all the opisthocons and all those protists. And so yeah, it's not bad. It's all right. It's pretty good. But yeah, so useful charts.
I'll check it out.
Everyone's saying great, thanks. Oh, you also have an invite from RJ Downard to his show Evolution Hour, which is on Wednesdays at 5 Pacific, right? It's 7 p.m. Central. What time zone are you in?
Eastern, so that'd be 8.
Yes. Yeah. So
they
go Wednesday. He would definitely love to have you. There's lots you guys can talk about.
I know what I should do. I should pitch my blog.
There you go. Yeah, exactly.
My blog is it's freethoughtblogs.com/fierce roller. So fierce roller is basically that's a nickname for Volvox.
That's awesome. I'll be sure to link that in the description.
All right, that'd be great.
After the video when I go back and do the little tags and stuff. And so I'll definitely link to that. So anyone who wants to go check out your blog can do that.
Cool, thank you.
Yes, sir.
Hey, it's been fun.
Yeah, also people have been commenting on your Volvox picture in the background. They love it. So
I just got I just got those made, and I'm probably going to be selling them soon. So keep an eye on the blog.
Well, there you go, Beej. There you go. You can buy the picture. So, alrighty then. Well, thanks everybody for coming, and thank you, Dr. Herron, for being here. It was very nice to have you.
It was great. I enjoyed it.
All right, everybody. Thanks for watching. You can stick around for just a moment.
Okay.
And all right. Well, thanks everybody. Bye.